So next speaker is Takuya Ito from IBM Research. Please welcome.
Thank you for coming. My name is Taku. So I'll present some work that I've done in conjunction with John Murray, and it's forthcoming in network neuroscience. How do I? Okay. So what is functional connectivity? So if you look in the literature, there's considerable variability in how it is used, from fundamental brain organization to identifying features for psychiatric disease, individual fingerprinting, and so on. But at its core, functional connectivity is just a statistical description. It is the correlation or temporal correlation between the activity of two neural units. And so if we were to lean into this, if it's just statistics, can we use an information theoretic perspective to try and help us interpret functional connectivity? So if you were to look in the literature in neural coding theory, in particular, they'd find a role for spiking correlations in information coding. So up here on the left is a paper, one of my favorite papers on this topic. It's a review paper by Marlene Cohen and Adam Cohen on measuring and interpreting neural correlations. There's also high fidelity coding with correlated neurons, information limiting correlations. More recently, papers on geometry of information coding, correlated neural populations, the structures and functions of correlations in neural population codes, and then one recent paper on trying to apply some of these ideas to MVPA analysis on pairwise, or MVPA analysis on fMRI data. So in general, these ideas from neural coding theory are themselves primarily statistical, and so it should follow that using these ideas could help us interpret correlations from fMRI data. So what is it? Overall, neural coding theory describes how something called noise correlations, which are statistically equivalent to functional connectivity, influences task information coding. And so what's critical about this is that interpreting a noise correlation strictly depends on understanding or characterizing its signal correlation. So the signal correlation refers to the similarity of task tuning curves between two neural units, or the similarity of their receptive fields. Right, so at the level of neurons, you would say these neurons fire to similar stimuli together, like line gratings or orientations. And at the level of brain region, you might have to zoom out a little bit and ask, do these two brain regions have similar cognitive functions, or do they have similar cognitive level receptive fields? And just to be clear about the analogy, noise correlations are statistically equivalent to functional connectivity, and signal correlations are conceptually similar to tuning similarity. Some people sometimes don't like these terms, but the naming convention is based from information theory or statistics, and there's a good textbook by David McKay on this. The general idea is that signal refers to the mean response of something across trials or conditions, and noise represents the variability of that response. So the intuition behind this is take two regions, a red region and a blue region. You can plot their tuning curves, so you have task conditions on the x-axis and their preference or their fMRI, the average fMRI response on the y-axis. But you can plot these two mean responses as a function of different brain regions. So you have two regions, parcel I and parcel J. And if you were to look across each plot, or each dot refers to a different mean response across a task. And then if you just correlate this across tasks, you can say something like the signal correlation here is about 0.8. Now, if you wanted to zoom in on just two tasks, a green task and a yellow task, and then try to analyze or understand them in light of their signal correlation axis, or the direction of their tuning similarity, you can say that, right, these are the mean responses of these tasks, and if you wanted to say that there's, the task FC was 0, you can look at the trial-to-trial or the moment-to-moment correlations of these two tasks. And then if you were to draw a hyperplane to decode these two tasks, you would say something like these two, having 0 correlations actually helps the decodability of these two tasks. Now you can say that if you induced a positive task FC to these two brain regions, you would see that these task correlations, because they're aligned to the signal correlation axis, they interfere and therefore are harder to decode. And then counterintuitively, you can think of if you induce negative task FC correlations, you actually end up having task correlations or task FC that are anti-aligned or orthogonal to the signal correlation axis, which therefore makes it significantly easier to decode. Now you can apply this to task, or in fMRI in practice, what you would do is oftentimes you measure task FC in relation to a baseline like rest. So you measure your baseline resting state FC, you make you characterize the task FC and say something like the task FC increases. So this is an aligned task FC aligned to the underlying signal correlation axis. And this would inhibit decoding versus if you have task FC that is reduced relative bit to baseline, you would see that it's easier to decode. And so in general, right, you can say something like when two brain regions have similar cognitive functions or a positive signal correlation, task FC increases would inhibit task decoding, while task FC reductions enhance task decoding. And in general, in a prior, anecdotally from a prior paper of ours, this is while I was a PhD student with Michael Cole, we found that across many tasks, you find that task FC primarily reduces relative to rest. What was interesting is that the reductions are most prominent within a network, and some might say regions within a network tend to have more similar cognitive functionality. Like two regions within a visual network likely have similar cognitive functionality. So how do we test this in practice in this paper? We take a multi-domain task battery dataset from King et al. 2019. This dataset has 26 different tasks, and so you can estimate the mean response across all of those 26 tasks, and then estimate or correlate them. So you can estimate the SC or the signal correlation matrix. So one thing to note is that the signal correlation matrix, generally speaking, looks quite similar to the network partitions. And then you can just look at the resting state, noise correlation, or FC, it's the same thing, or the task and see, and then again the difference. And again, you see reductions, which is consistent with the prior slide. So how do you actually make inferences on these two matrices, right? Again, to understand how task FC influences its coding properties, you have to take it in conjunction or characterize it in conjunction with the signal correlation matrix. So what you can just do is take this matrix on the left and take the second matrix and then compute the element-wise multiplication. It tells you something about whether or not the signal correlations are aligned with how the noise correlations change. So regions in red are aligned, which would inhibit coding, and then correlations in blue are anti-aligned, so that would enhance coding. If you wanted to take a more narrow view, just look at which networks have which types of correlations, you'd find that in general, most networks have this anti-aligned correlations that enhance decoding. But one region in particular, the default mode network, has mostly red or mostly aligned correlations, which appear to inhibit coding. And then you can just test these ideas out, right? So you identify a clique or a set of regions that have either aligned or anti-aligned correlations, do a decoding analysis where you decode the tasks or the multitask decoding analysis. And then you'd see very clearly that regions that have anti-aligned correlations with each other have significantly greater decodability relative to aligned correlations. So all of that just to say that in principle and empirically, neural coding theory holds in fMRI data, and it provides a theoretical framework to interpret how these FC changes influence task coding or task decoding. Just to wrap up, again, functional connectivity can be analyzed from the perspective of neural coding theory, which aids in our interpretation of FC changes. And again, I highlight this paper if anyone's interested in the topic. And what's important about this is understanding how task FC changes requires knowledge of the signal correlation, the tuning similarity. Right, so when task FC changes are in the same direction as the signal correlation, this inhibits task decoding. And empirically, we find that most signal correlations, or most regions are cognitively similar to each other, are positive, so it would follow that most task FC decreases would enhance decoding, and FC increases would inhibit their decodability. Right, so these are just the matrices we found in the paper. And then just as a final comment, what this would imply is that correlation increases do not strictly imply communication increases. What you have to consider is the correlation changes with respect to the signal correlation. That's all I have. Thank you.
Is there any question on the mobile app? I couldn't find one. So actually, I have one question. Great talk, by the way. So a question is, would you expect to see like some variance across different task domains, especially for like a normal like a cognitive task versus a moving watching task?
Yeah, so in general, right, so in this data set, we try to do as diverse a cognitive battery as possible. So this is the data set that was publicly available. But in principle, I think these ideas would apply if, for example, you just want to study vision. You can look at conditions across different types of visual conditions and characterize it there. I think it'd be harder to make inferences if you have kind of binomial or like a bipartite sampling of your tasks. So, yeah.
Okay. Thank you. Let's thank the speaker again.